sludges for biohydrogen producing with dark fermentation. Thank you for your invitation and for being here to stand here for 2 days after the launch. So I hope this presentation is going to be interesting for you. My name is Miguel Ángel Sánchez Gaton. I'm a researcher of the Cartier Foundation, and I'm here to present a work together of our area that is focused on the anaerobic digestion of different industries, always focused on the production of hydrogen, biohydrogen, through the dark fermentation.
Por si no lo conocéis, es un centro tecnológico.
So if you don't know this Cartif, is placed in Boecillo, is focused on the work of research and towards the companies as the main focus in different fields. Let's start. We wanted to start working with the dark fermentation as a very promising one that Cartif is believe in to produce biohydrogen as an alternative biological process. It's called dark fermentation because, of course, it's produced without lacking the light compared to other technologies that are already existing and they use light. What are the products of this process? Is mainly hydrogen and methane as well, and CO2 and some VJN. This technology is in a maturity level that is like in the middle, 4-5. And this some of the challenges through the research, I think, the VFA, we could overcome. For example, the pH instability during the process and the variability of the inoculum. In this second challenge, we have focused a lot, and we present in this Congress just a small part of it, but this study is within a very wide work where we have been doing a very deep analysis from a taxonomical and microbiology point of view. But here we have only focused on the performance of the biohydrogen production. So we're going to study even 5 different anaerobic digesters from 5 different industries. Thanks to some of the partner collaborations in our region, we could take some samples from 5 different industries. Here you can see a sample from the city and also digested from a sugar company and from the meat industry and cellulose industry and the bread industry. We tried to cover a wide spectrum about the digested products, and then we took some, created some trials from 1 liter, so we are far away from the market. But we were able, with this 1 liter sample, and select and identify the best inoculums, the best digesteds, focus on the process of biohydrogen in the dark fermentation to produce this biohydrogen. That's the goal, to select which one is the best. Here we have an experimental methodology that is quite clear. We want to characterize the digested. We want to do a pretreatment of an acid pretreatment to balance the pH to 5.5. We don't want to study how is the influence with different pHs, but we want to keep it fixed. And also we do anaerobic fermentation trials with glucose. We feed it in a limited way, so the process, in like batch process, can be able, this organic matter can be able to be consumed totally. In order to compare this, the potential of each of the studied inoculums. Based on those trials, we have analyzed the production and characterization of the produced gas, mainly hydrogen, but we also have CO2 and methane. And also we have done a microbial analysis to see how these microbes have varied depending on the inoculum that we are using. The conditions are the same for all of them. As I said, we have used 1 liter reactors. We have done these trials 3 times, each of them, so the precision was very suitable. We have done a acid pretreatment, acid pH pretreatment with a mesophyll regime and with an orbital agitation. All of them have done in the CAR-T facilities, in the labs, and we have tried to use different technologies that are available to be able to carry out all this to get these results. Let's move directly to the results. This is the most important part. We can see here how they behaved, all these inoculums that we have done in the trials. This you can see in the graphic, the gas production from the gas mix, not CO2 and hydrogen, but like the mix. So based on the results and the register of the produced volumes during the process, these are very short processes, in this case with a maximum duration of 100 hours. And we were able to model from the Womper modified formula the curves that are presented in the dark fermentation of each of the inoculums. The result is quite clear. We were able to see that the inoculum coming from the meat industry that is fed by protein, high protein concentration, it produces gas much more than other waste. You can see it here in the graphic. It's true that globally, the waste coming from the sugar company manufacturer is producing many more gases, but it's focused on the biohydrogen. The difference is very significantly. The meat inoculum is the one who is going to be the most representative process of dark fermentation, and also the one that it could be more suitable for this process. Here you can see how it was modified in the mix, the reactor mix. We started from a 4.5, 4.6 pH in this mix. And depending on the inoculum, the ones coming from meat and bread and the urban one, the pH has dropped. So it is more acid than the initial ones. That means that the process is helping the production of hydrogen, in the opposite of the processes that we have seen that there is an increase of the pH. Sometimes it was moderate or even high. The process was directed towards the methane production instead of the biohydrogen production. At the end, we have these results, preliminary results that we obtained, and we wanted to see what was happening inside this reactor from a microbe point of view. We wanted to analyze the 5 inoculums before and after the trials and see what communities, what phylum, what gender. We had like a box with the different small boxes inside, and the phylum had different classes and it has different genders. And we wanted to see which genders with microbial communities were able to adapt better to these processes and to see the result. We see there is a very clear process for each of them. For example, for the inoculum coming from the wastewater treatment plant, we can see the development of the lactobacillus leptococcus that are associated to the hydrogen production that is low or moderate. This slide had some animations with the idea of associating each of the graphics with each of the inoculums, but I see it's not working. I will try to explain it in the best way. The sugar manufacturer has an inoculum that is orientated based on the characteristics towards the methanogenic taxons growing. So we have high concentrations of methane. So it's a very interesting process for this study. We are going to classify it as less suitable inoculum for this process. The winner, of course, was the inoculum coming from the meat industry, highlighting the great growing of the Costribia class. As you can see in this process of growing process, was focused during this growing was on producing hydrogen compared to the other bacteria. When we do this pH adaptation, we are simplifying the microbial communities in order to for the communities that can produce biohydrogen, for example, from the meat industry, they could leverage the protein content with this inoculum in order to produce a higher volume of biohydrogen. And then when we move to the cellulose industry, we have the bacilli class. It is dominant within this microbial community and is obtained with a methane concentration level that is low, let's say. And the digestate coming from the bread industry, we achieved values like of hydrogen that are like moderate values of hydrogen. At the end, what we can take from this work, I remind you that it's preliminary. For next year, we hope that it's done. Next year, we will present all the results that are going to be more specific. But we were able to understand that the digestate coming from the meat industry could be the best inoculum possible, at least for trying to boost the development of this process oriented, focused on the biohydrogen production. The acid treatment, the previous acid treatment is very useful, but it depends on the digestate that we are using, okay? So from Cartif, we think that at the end, the process of dark fermentation could be promising. But we should not lose the focus that this technology is well matured, but at a lab scale can be done only with much wider studies and pilot samples that it could allow us to go towards a more like market size. I think I'm not on time a lot. Thank you very much, and I hope it was interesting for you. Thank you.